when it's your turn. Okay, so good morning. I'm Nerea Piñeiro, representing OYSTER as well as Dinesh. One of the activities that we in OYSTER are more proud of are the early career researcher projects that we started in 2022 with a pilot call. The idea of these projects is to give early career researchers the opportunity to be the leader of their own projects, as this is something that many times are asked for to prove our leadership in postdoctoral goals, but there are very few opportunities for us to actually get funded. So what we wanted to do was to provide this opportunity, but as well to provide cooperation between different EuroMarine institutions. With this in mind, we opened a pilot call in 2022, and we have 15 projects, and we were able to fund one. We were very happy with the results, so we decided to open a second call in 2023, but this time, thanks to EuroMarine and their trust in us, we were able to fund 2 different projects. And today we are going to do 3 small presentations of the awardees of this project, so they can tell you about the projects, and you will know a bit more where your money is going to. We hope you are as proud as we are. So the first project we are going to introduce is the one that was funded in the pilot call by Pavanee Annasawmy, which is the PI of this project, the NEET project. She has the floor.
Thank you. Can you hear me?
No.
No? Oh.
Yeah, you on the top. Yeah. Okay.
Yeah, we can hear you, Pavanee. You can share your screen.
Great. Can you see my screen?
Yeah.
Okay, great. Well, thank you, everyone, and I'm sorry I couldn't join you in person this time. So my name is Anjali. I'm a postdoc, and so this project was actually the idea came when me and the 2 other ECRs, Arthur and Guillaume, we were on a cruise ship, collecting data for our respective projects. And while talking with each other, that's when we realized, okay, there's a knowledge gap, and how can we? I mean, we just saw like opportunities for us to work together. And the idea for the project actually came before the ECR pilot call. So this call was actually, it really came at the right moment for us, because it allowed us to concretize our idea in some way, to make it more solid, and to actually work on the project and get funding to work on this project together. So this project is called NEET, and we're looking at MicroNekton diversity, distribution, and abundance using 3 different techniques: environmental, DNA, acoustic, and trawls. Okay, so as a bit of a background, MicroNekton are organisms which are of 2 to 20 centimeters in size. They consist of crustaceans, cephalopods, and mesopelagic fishes. And compared to plankton, MicroNekton are able to swim against currents. So these are not just passive organisms which drift with currents, they actively swim, and they actively also migrate. So some of these organisms, most of them live in the deep ocean, and at dusk, they would migrate up to the epipelagic zone, and they would migrate back to deeper waters at dawn. But typically they would live in the mesopelagic zone. So that's why they're mesopelagic organisms mostly, for most of them. And MicroNekton are very important because they have several functional roles. They play a key trophic link between lower trophic levels. They connect zooplankton to higher levels, to tunas, for example. Tuna would typically feed on MicroNekton. And they also, by migrating, so up and down the water column every day or every 2 days, they contribute to the nutrient recycling, and they actively play a role in the biological carbon pump, because they would kind of take up the carbon at the top of the ocean, so in the epipelagic zone, and they would bring it down to the deeper ocean. So hence they play a key role for us in fighting climate change as well. But however, they're starting to be we're starting to have huge exploitation interest because there's the estimated biomass is in several billion tons. So MicroNekton represents an untapped resource. So there are several countries which are looking into exploiting MicroNekton either to do nutraceuticals, as fish meal in aquaculture farms, or maybe even for human consumption or to make dietary supplements. But despite our exploitation interests, there are still significant knowledge gaps. We still don't know the exact biodiversity of MicroNekton, and that's because, well, you see later on, because of the tools that we're using, we're not sure of, we're still discovering new species, actually. We're not sure either of the taxonomic identification. That's so every year new species are being reevaluated and taxonomically re-identified as well. We don't know much about their life cycle because these are deep sea organisms, and there's no way that we can know their full life cycle. We're still discovering the habitat ranges. So even though we know that they live in the mesopelagic zone, some of them, new studies are telling us that maybe some do live in the bathypelagic zone, or maybe some don't exactly live in the mesopelagic, but more in the epipelagic. So we're not sure actually. And the abundance and biomass, again, we have estimates, and these estimates range by several orders of magnitude between studies. So there's no consensus. We know nothing of the reproductive patterns or the trophodynamics. So there's still a lot to be discovered. And that's despite the crescent threats that MicroNekton are facing. New studies are telling us that MicroNekton, they are sensitive to plastic pollution. So some of them have plastic particles in their stomach, microplastics. Modeling studies tell us that MicroNekton could potentially be impacted by climate change, deep sea mining, and also fishing incentives in the future. So our poor knowledge of MicroNekton communities comes from the limitation of the tools that we use traditionally to study these organisms. So traditionally we use nets, trawl nets, but these trawls, they capture MicroNekton differently. So and organisms can escape or avoid the nets. Trawl nets are also time consuming. And when you go on multidisciplinary cruises, you don't always get to do an unlimited amount of trawls. It also depends on the objectives of the cruise. And if it's a cruise where there are varied objectives, you don't get to do that many trawls. And also trawl nets provide only a snapshot of the communities present at that time of day, at that depth, and at that spot. So you can't really extrapolate to other areas because it's just at this point in time. Traditionally, we also use acoustics. So these acoustic, it's active acoustic because there's typically an echo sound on the hull of the ship, and it sends electromagnetic waves in the ocean. And once these waves hit the swim bladder of the fish, the signal gets reflected back. And we can then look at, so this is typically acoustic data, where it tells you the intensity of MicroNekton densities. So in red means higher densities, and in blue means lower densities. So there you can see the migration of MicroNekton, so moving up and down the water column. But as you can see, we can really get a density. We can't really say if there's more fish in there or more crustaceans or cephalopods. With acoustics, we can't really resolve the taxonomic composition of these MicroNekton. So the new approach that we're starting to develop is using eDNA. So eDNA is used to primarily identify species that are present in the environment, and it has a wide range of applications to detect target species, but also to as a inventory of the local biodiversity. And that's because we can eDNA, we can use this technique because organisms, they would shed DNA in the water column. So either through blood or through tissue samples, through skin or gamete release. So by shedding the DNA, and if we collect this water, we can then try to detect the organism that was present. So the advantage of eDNA is that it can sense what has been there. It can detect cryptic species, but there are several bottlenecks still that we have to resolve, and that's because it's a fairly new technique. So eDNA relies on reliable reference libraries that do not currently exist. So we're currently building them. And you'll see later on how NEAT is contributing to building these reference libraries. And also eDNA relies on net sampling to ground truth. So you also have to go back to the trawls. And it's not quantitative yet. Some organisms shed a lot of DNA, some much less, and eDNA can get degraded in the environment. So for the time being, we can't really use eDNA to say there's as a quantitative analysis. And sampling factors as well, since it's fairly new, we don't know how much do we sample, how much volume of water to best capture these organisms, and what primers will we use to best capture these organisms. So prior to starting this study, I thought this has been resolved, but not yet. While discussing with people in the community, there's no consensus actually about primers, which primers to use to best capture MicroNekton, since, well, we're fairly new, like starting to develop this and study this. So now for the sample collection study sites. So the data comes from, so like I mentioned, a cruise that we did, the ECRs in the Indian Ocean, so the Mozambique Channel, between Africa and Madagascar. And also from a study site around Réunion Island. Guillaume took part in that cruise and he collected all the eDNA samples during that cruise. So NEAT, the project combines traditional tools, so trawls and acoustic and eDNA. And for the active acoustic, we're developing, so I was developing an algorithm as part of my current postdoc that I'm applying to this project to try to classify the acoustic data. So as I mentioned before, we can only get densities, but from that density, I wanted to try and classify the density into different groups. So if we find more fish or gelatinous or crustacean. And we're also testing different eDNA collection techniques because we're not sure which one would work best for MicroNekton. So eDNA was collected from water, filtration of water coming from Niskin bottles, CTD bottles. And also we collected water at the end of the trawl net. And we used a new passive eDNA collection method called a metaprobe. So this metaprobe is just pieces of gauze, which was put into a 3D printed plastic collection device. And this metaprobe, typically you can put it everywhere. Like you can put it in here we put it on the trawl net, but we also tried to put it on a CTD and yeah, just explore different methods of using this metaprobe, this device. So the objectives of the project was to determine which eDNA sampling technique best captures MicroNekton, to identify which markers best capture these organisms, to barcode the specimens collected in the trawl so as to enrich the DNA reference databases, to complete the taxonomic identification of the samples collected in the trawl, and to standardize the acoustic algorithm to be able to detect MicroNekton acoustic groups. And finally, to integrate all these techniques, eDNA, acoustic, and trawl, to investigate the diversity, distribution, and migration patterns of MicroNekton. So originally, this was the Gantt chart, the work plan that we had. And I'll show you later on where we are right now. So for the methodology for the eDNA, so we did a filtration of the samples on board. We stored it, and then afterwards you'll have to extract, purify, and amplify the DNA and do the sequencing of the DNA and later bioinformatics. So Guillaume last year, he tested a lot of primers to see which one would work and which one would not. So I won't go into details of these, but these are quite a lot of primers that he tested, and the results were quite mitigated because with the primers that Guillaume used, we identified a small proportion of myctophids. Typically, these are the most common genus found, and in this area, typically there would be loads of myctophids, but we didn't identify that much myctophid. It's a really small percentage compared to other fish species. So now what I'm doing is that I'm testing other primers, so these two. So MyFish 12S, I discussed with people in the community, and a lot of them advised me to use this primer. And a few labs advised me to use this primer to detect crustaceans and cephalopods. So right now, these samples are being analyzed in a lab in France, in Montpellier, to try and see whether we would detect more MicroNekton if we change primers. So now for the methods, so for acoustics, so like I mentioned, so typically the echo sounder would send sound waves in the ocean. And so we can determine the strength of the echo by calculating the time that it takes for the wave to get back, so we can know the exact position of the shoal of fish in the ocean. And also typically we would use, so this comes from literature, so a lot of studies have shown that so acoustic data can be collected with a variety of frequencies. So frequency is just different lengths of sound waves that we would send in the ocean. So these are the frequencies that we use typically. Well, they're quite a lot of frequencies and typically smaller organisms. So we could detect them with higher frequencies, and large organisms such as fish and squids, we can detect them with lower frequencies. And this is the basis of the acoustic algorithm that we identified. We exploited this how do I say, this theory that small organisms would respond to higher frequencies and large organisms would respond to lower frequencies. So with that in mind, that's how I develop
Excuse me,
a model. Yes?
Angeli, we need to keep going to the next presentation. Could you go
Okay.
to conclusions? Yeah,
Yeah.
sorry.
So I'll go to conclusions then. Thanks, buddy. Okay, these are just the organisms collected in the trawl and that we're identifying them. Okay, so the next steps for this study is to standardize the algorithm across sites. And in red, you can see the steps that we have completed already, and in violet, blue, and gray are the steps that still need to be completed. So we have to standardize the algorithm, complete the taxonomic identification of the organisms collected in the trawls, add the genetic sequences of all specimens that we barcoded to online reference databases, and finish bioinformatics of all samples. So the final steps would be to compare the number of reads obtained from each eDNA technique, sampling technique, and from each primer to be able to say which eDNA technique works best and which primer works best to detect MicroNekton. And ultimately to calculate the biomass at each site. So using the trawl data and using acoustic data, and also calculate the abundance at each site using trawl data again and acoustic data, but also using eDNA for the number of reads and the acoustic data. So to be able to say at each site how much MicroNekton we have and how much MicroNekton groups we have. So thank you, and sorry for the long presentation.
Thank you, Angeli. We are now going to the next presentation. We are going to have two presentations about the projects funded in 2023. And the first one is the EMFish Project by Lotte Dahlmo. Can I just move the computer a bit closer? Okay. Okay. Perfect. Hi, everyone. Very exciting to be here. My name is Lotte Dahlmo, and I'm a PhD candidate at University of Bergen and at NORCE, located both in Bergen in Norway. And let's see. Yeah. So today I'll be presenting our EMFish project that was funded by and supported by EuroMarine and the OYSTER group through their ECR cooperation project. So we applied this fall and got the funding and approval right before Christmas. So this is going to be a presentation about what the project will entail. And through the project, we are investigating the impact of electromagnetic fields from offshore wind farm power cables in the North Sea on the endangered spiny dogfish using novel acoustic tags.
And just to give you a quick introduction who we are three people, three early career researchers. Two of us are both named Lotte. Lotte Dahlmo is the leader of the project. However, both Lotte and David are both unavailable to attend this Open Science Day due to other obligations. So luckily for me, I was able to come instead. Lotte, she works at LIS together with David, and they are both, or we all are, very interested in the marine environment and fish ecology and tracking. And as I mentioned, I'm at the University of Bergen and at Norse, and we all have experience with tracking fish and sharks. So they contacted me in the fall, said that they had an idea of investigating electromagnetic fields from offshore wind farms, which are being installed, especially in the North Sea. And then they contacted me because I have a lot of experience with acoustically tagging spiny dogfish through my PhD project. So that developed the EMFish Project. And a bit about the background. Offshore wind farms, you've probably seen a turbine of these, both on land, but also now in the sea. And the North Sea is an area with a high density of the wind turbines. But we don't really know the effects of them. We need to move towards renewable energy sources, but renewable doesn't necessarily mean sustainable. So this is the area in Europe. And then we have the shark species, the rays and the skates especially, are probably impacted by the electromagnetic fields created by the power cables from the turbines and the offshore wind farms, because they have receptors in their skin that can detect these signals, and they usually can use it for identify prey or avoid predators or for navigation and orientation. So when we install all of these offshore wind farms, it is likely to that it will affect their behavior. So this is parts of Europe. This is the area where we're studying. The green areas are the areas where the offshore wind farms are already installed with nearly 400 turbines. And then the gray shaded two areas are where they're planning to install more offshore wind farms. And then there are cables from each turbine, and then there are fewer export cables through that goes to land that exports the generated electricity. And these are the cables that create electromagnetic fields, which is the basis of our project. And we are relying on a methodology called acoustic telemetry. It's a way of tracking aquatic animals, where we use these small tags that transmit sound signals. And the sound signals are unique so that we can identify which individual it was. And these sound signals are then stored by these black receivers that are deployed in the water, and they are stored on those, and we can download the data and look at what time and date an individual have been around and been detected. But what is also cool with acoustic telemetry is that we can have sensors in the tags. It's common to use depth sensors or temperature sensors. So there's also more novel sensors like acceleration and predation. And now one of the manufacturers, Dahlmo Biotel, which is a Norwegian company, they are developing a sensor that can detect the electromagnetic fields. So our project entails trying these tags. which is the first work package. Then we will track spiny dogfish with the tags. And lastly, we have a social outreach work package because we find it very important to communicate science not only to scientists, but also to locals and kids and industry partners and governments. The first work package is the actual testing of the tags. When we have new equipment, new technology, it's very important to actually validate and test it, not only in lab settings, which is often where equipment might be tested, but it's also very important to test them out in field where it will be used. So through this work package, we will create baseline field data so that it can be used for future studies. The second one is the tracking. Sharks have important roles in the ecosystem. This is me tagging one of my 82 dogfish for my PhD project. They are very easily found if you find them, put them in groups. If you don't find them, you don't find them. Yeah, I tagged like 32 one day, and the next day I tagged none. So it's a very up and down. But yeah, we bait them, we angle with a rod, with baits, and then bring them on board. Quite easy. Last one is the social outreach. This is the draft of a scientific illustration we are planning to make. It's going to be kind of meant to be used across borders, and will be primarily targeted to kids, but also used in settings like these. And we will create like handouts for museums and aquariums. If anyone have any contacts that might be interested, please come find me. And then plan forward is to get the tags, hopefully within a few months, and then do the actual field work. It shouldn't take a lot of time to do the actual field work. Yeah, finalize the scientific illustration. Also got some funding to have a social outreach event in Norway that I've called Shark Summer, bringing some kids out to the fjord and learn about fjord ecology, where this is a perfect project to tell them about. And that was it. And again, thank you for EuroMarine and OYSTER for funding the project, for bringing me here. And I'm very excited to be here and chat with you guys.
Thank you, Lotte. As we are a bit late, questions you might have better in the coffee break, I'm sorry. So we will go forward with the last project, the Argyro Adamopoulou. Is that correct?
Yes.
She's going to tell us about the Plast Project.
Good morning. Thank you also for funding the project as well. I'm very glad to be here, and I read in Wikipedia that Bologna University is the oldest in Europe, so it's also very nice. And together with Giuseppe and Marianna, that we wrote together the project, we were very happy to receive the grant. So the project is called Harmonized Spectral Analysis for Environmentally Weathered Plastic. All right, so plastic is a quite fantastic material. I mean, we all use it daily. It was invented in 1907, and its mass production started in World War II and in the 60s and 70s when we use it for plastic, for packaging, food packaging. Plastic is very fantastic because it's used for an extended variety of products, and also it endures and it really persists in the environment. For example, a plastic bottle can take from 450 up to 1,000 years to be decomposed. Annually, 400 million tons of plastic are produced, and from them it's estimated that about 14,000 million tons of plastic are end up in the ocean. So you can imagine that approximately what has been produced so far, it exists still in the environment, even in the form of micro or nanoplastic. So plastic, when it enters in the environment, in the terrestrial environment or in the ocean, which is our focus, it fragments into smaller pieces. In our case, it fragments into microplastic, mesoplastic, and macroplastic, which is the fragmentation range we are going to investigate in the project. And monitoring microplastics worldwide, there are many different methods being used. So there is a need to make a more harmonized method to monitor microplastics. So in this photo, you can see some microplastics. These are about 1 millimeter and a little bit smaller. You see the scale. The photo was taken from a stereo microscope, and these microplastics were collected with a mantanet, which is a net collecting sea surface floating microplastics. So when we are monitoring or investigating microplastics only visually through a stereoscope, this can yield to false positive and or false negative results. So it is really necessary to use polymer type identification in order to confirm that particles that we have classified visually as microplastics are indeed plastic polymers. So it's really necessary for all the scientists using monitoring plastics and using working on microplastic pollution to use common and harmonized polymer type identification methods in order to improve data consistency. So FTIR spectroscopy is commonly used for polymer type identification. FTIR produces an infrared spectrum, which is the fingerprint of the polymer type of the identified plastic. Here you see it's at the top corner, you see a microplastic, which it happens to have the heart shape. And you see its fingerprint, so the infrared spectrum, which is a polyethylene. Polyethylene is the most common polymer type we find at the surface floating, and it accounts for 80% of the plastic we find. And polyethylene, it's the polymer type that's used mainly for single-use plastic. I see also a lot of plastic bottles. I hope, well, this presentation to affect you and try to use less and less plastic when we can avoid it, of course. So all of your plastic bottles that I see here, they have this fingerprint. So it's the importance of polymer type identification is to make sure that what we see is to really identify the plasticity. So the spectrum is a source of information, which serves as polymer type verification tool, and additionally, it can be used to distinguish between natural and anthropogenic polymers, and pristine and weathered plastic. And also it can indicate the origin of plastic. When we say indicate the origin of plastic, it can show us also for example, if I know it's polyethylene, then I can estimate that probably it originated for a single-use plastic material. So it can also help us to alter the legislation when we know the exact polymer type of the plastic. And when we say weathering process, well, weathering alters the polymer spectrum. And by defining the spectrum alteration, we can define the age of the plastic. And then we can avoid spectrum misidentification. Defining the age of plastic is quite important because then we know for how long the particle that we identify has been wandering in the ocean. So our goals as a Plast Project is an intercalibration exercise among the collaborating laboratories, which is in Greece, HCMR, in Italy with Giuseppe, and in Belgium, in Greece with Mariana. And there we aim to identify and validate the polymer type of floating microplastic, mesoplastic, and macroplastics collected after a flash flood event this August in central Greece. I don't know if you have seen in the news, we had the major flood around Volos area. And this, of course, brought huge amount of plastic in the sea and in the bay. So through the intercalibration, we are aiming to promote reproducibility and data consistency. And then more goals of our project are to develop a method that will optimize spectral quality assessment, validate polymer identification, and estimate plastic weathering status using R. This method will be uploaded at OpenSpeki, which is an open access tool already used. Furthermore, the intercalibration spectra will generate spectral libraries for environmentally weathered plastics in order to advance the OpenSpeki existing reference libraries. So this libraries generation will further contribute to library harmonization, which is it's really critical. And I don't know what I saw from the presentation of Angel, it's like really crucial to have correct reference libraries. So the end goal of HardPlast will be to contribute to data accuracy, quality improvement, comparability, and reproducibility of plastic particle identification and weathering status estimation. Thank you on behalf of Giuseppe and Mariana. We're very thankful, and thank you for inviting us in Bologna.
Okay, thank you. That's all the presentations we have today. I would like to thank EuroMarine for allowing us to carry on with this idea, because we think it's very necessary that early career researchers have the opportunity to get independence and their own funding and to try new ideas. And sometimes it can be very frustrating when they are asking you to provide leadership, but they are not giving you the opportunity to actually make mistakes or to try. And we think that EuroMarine, as they said before, they are trying to fund things that not other people are funding. So this is a great thing, we believe. And we are very happy to continue with the project. So thank you so much.
Thank you. There is just one question in the chat for Argyro. I don't know if you have the time to take it.
Excuse me?
There is just one question, or rather a suggestion for Argyro on the chat. So maybe you can have a look. So thank you, Argyro, for your talk on a fascinating project. I would just suggest the author to check the published database of Robin Lens. So it's basically a suggestion and a well done.
Okay, thanks. I think we break for 20 minutes now because we want to change the agenda.